RNAi or RNA interference. Now, this RNAi is actually cell's built-in immune system. This is the process when cells they make small RNA molecules that will turn off specific genes or your targeted mRNA. This RNAi works by recognizing unwanted RNA molecules and shut it down before it can create any damage. This used to defend against viruses and transposon. So you can see that RNAi is a natural process which is originally evolved as a defense system. In research, we use RNAi to knock down a gene, that is, to disrupt the activity of a gene so that we can study its function. Now, how this process exactly works? The cells actually, on sensing any unwanted RNA, they make small RNA molecules called siRNA and miRNA, which act as guides. That is, it directs one protein complex named RISC or RNA-induced silencing complex that could bind to and cleave your targeted mRNA molecule. This is the principle behind RNA interference. Now, the important requirements for RNAi are as follows: double-stranded RNA, dicer, and RISC. The double-stranded RNA, it can come from virus, from transposons, or from outside, that is synthetic double-stranded RNA. For some time, the single-stranded RNA are being made inside cell during normal cellular process. Then they can fold back on itself to form the double-stranded RNA. Or sometimes they are acted upon by RNA-dependent RNA polymerase to form double-stranded RNA. Now, on sensing double-stranded RNA, dicer gets activated. Now, if you notice, this dicer actually belongs to RNase 3 family, which is an endonuclease. So this dicer is actually responsible for cleaving those long double-stranded RNA into small siRNA. Now, if you see, this dicer actually contains two RNase 3 domains that possess the endonucleolytic activity of dicer. There are two types, RNase 3A and RNase 3B. So how this operates? Now, on sensing the double-stranded RNA, the PAS domain of dicer, they get attached to the 3' side of that double-stranded RNA, and then 21 to 25 nucleotides away from this binding site, the RNase 3 domain or the RNase 3 enzymes, they cut. Now, if you notice, the RNase 3A and RNase 3B, they cut on opposite strand. That is, if RNase 3A cut the top strand, RNase 3B cleaves the bottom strand. Now, this cut do not exactly occur on the opposite side. Instead, there is a 2 nucleotide offset, and this what generates a 2 nucleotide overhang at the 3' side of your siRNA. So the long double-stranded RNA, which is around 100 to 1000 nucleotides long, they are being cleaved by dicer to form siRNA, which is around 21 to 23 nucleotides. Last component is RISC or RNA-induced silencing complex. Now, Argonaute protein is the most important component of this protein complex because they have endonucleolytic activity, which is responsible for cleaving your target mRNA. Now, let's understand the process. Now, when the double-stranded RNA is being introduced in the cell, dicer acts on it and cleave it to form small siRNA molecule. Now, these siRNA are double-stranded. While loading to the RISC complex, one of the strands of your siRNA gets degraded. That is called the passenger strand. And another strand survives. That is called the guide strand. Now, as the name suggests, the guide strand. The guide strand directs RISC to bind to the target mRNA. How? Well, actually, the guide strand is complementary to your target mRNA. Hence, they base pair. And once base pairing is done, the Argonaute protein, which possesses the endonuclease activity, that cleaves the target mRNA. The cleaved mRNA fragments are further degraded by the exonuclease in the cytoplasmic P bodies. And that's what causes your gene silencing. Now, let's understand a bit more detail how the Argonaute protein actually works. Now, this Argonaute protein contains one important domain called PIWI domain, which possesses aspartate histidine residues in its active site. Now, this amino acid residues recruits magnesium ion. Now, this amino acid residue, along with the magnesium ions, they pulls proton away from the cytoplasmic water. Now, this cytoplasmic water, they are normally present in the cytoplasm. Now, once they pulls the proton away from the water molecule, those water molecules become extremely reactive. That is, they act as a nucleophile. Those nucleophiles then make a nucleophilic attack on your targeted mRNA. Now, once the phosphodiester bond in your targeted mRNA is broken, the mRNA gets cleaved. And these fragments are further degraded by the exonuclease and your gene silencing occurs. Now, this magnesium ion plays a very important role. Now, during the nucleophilic attack, actually, some extra negative charges are developed on these oxygen atoms. And magnesium stabilizes those extra negative charges that are developed, which helps to carry the reaction forward. And this is the basic principle how RNAi normally occurs.